thank you everyone for taking the time out of the morning to hear the update from DelMar Pharmaceuticals. My name is Jeffrey Baccia. As Brendan said, I'm the president and CEO of the company. I'm joined by Dennis Brown, my co-founder and our chief scientific officer here today, and we look forward to a productive day. I see several familiar faces and some new faces in the audience, so again, thank you very much. DelMar, as was introduced, is a pharmaceutical company focused on oncology. We are just about to cross our first year as a public company, so our 1-year anniversary from becoming a public company. We did go public through a reverse takeover last year in the first quarter, which requires us to be on the OTC bulletin board for 1 year. And as we come through that seasoning period, we are looking at strategies certainly to uplist to more senior exchanges. Our safe harbor statement is here for your reference. Our mission as a company is to really develop and commercialize products that we would consider to be well-validated cancer therapies and plug them into indications where the mechanism will solve an unmet medical need. When we talk about well-validated, we're focused on assets that have been studied previously primarily at the National Cancer Institute, so we have a good understanding of the mechanism, understanding of hopefully some phase 2 activity, as well as the toxicity profile of the asset. And as I said, we study that and we look at the landscape of modern care and plug it in where it makes sense and run as quickly as possible. In terms of this asset and our company and accomplishing this mission, as I said, our lead drug candidate is something we consider well validated. There are over 40 clinical publications from the original NCI work that gives us direction to build from. The management and the rest of the team has a strong history of product development as well as product and market exits for investors. Interestingly, this asset is also what we learned approved in China. I'll talk a little bit about that, but there's some blue sky opportunity in addition to our focus in the development of the product in the United States that we can tap into. Our development plan, because we are focused on a refractory disease and a relatively rare cancer, an orphan indication, is relatively streamlined and straightforward, and I'll talk about that strategy. And we have cash on the balance sheet through the first quarter of 2015, which means we've got some runway in order to accomplish those goals and build value. We've raised about $13.5 million to date, including that RTO round. And as I said, we've got cash on the balance sheet for through the first quarter of 2015, which is a nice way to say that a little bit later this spring we'll have about a year of cash on the balance sheet, so we're actively looking at opportunities to shore up that balance sheet. Our capital structure gives us one of those opportunities. You'll see we've gotten about just shy of 60 million shares outstanding on a fully diluted basis. A handful of the warrants associated with the company were issued to the investors in the RTO round, the going public round. And we can call those warrants based on certain trading parameters. That would bring another $10.5 million of paid-in capital into our company and give us enough capital to bring our lead program in refractory glioblastoma to approval at the FDA. And we think that's a nice position to be in. We've modeled the capital structure on other companies that have done this successfully, and so we're very pleased to hopefully move forward and accomplish that as well. Our goals for 2014 are twofold. Number one, advancing our lead program in glioblastoma into a registration trial. This is what we believe is going to be the biggest value driver, and we're on track to initiate that registration trial in the second half of this year. The other opportunity that we believe is that because the product is commercialized in China and because we have new data that can help to reposition the product through a partnership, we can help to capture some of the value that we'd be creating in the market where the drug is already approved. This next slide is some of the highlights from last year. It's very busy, it's very hard to read. The whole point is we've been very active in moving our business plan forward and communicating with the marketplace since we became a public company, and we'll continue to do that. We're very open to communicating directly with investors as well as through the appropriate broad channels, and we'll continue to do that. I'd like to focus now a little bit on our first product opportunity, VAL083. This program is currently in a phase 1-2 clinical study, which we are advancing toward completion, and we expect to be completing that study and, as I said, moving into a registration trial in the second half of this year. There are a lot of programs in development for glioblastoma, because as I'll talk about, it's a very serious disease that has huge unmet needs. This is a unique program because in comparison to the other advanced programs in glioblastoma, this is a small molecule chemotherapy. It's not an immunotherapy, it's not a vaccine, it's a very simple molecule that binds to DNA, interrupts the cell division process, and leads to cell death in the cancers. There are over 40 clinical publications around this drug in various tumor types. The drug was originally studied here in the United States by the National Cancer Institutes in what we would call the original war on cancer, the late 70s, early 80s, where the government was funding a lot of work to try to cure cancer. And so the NCI was throwing basically whatever they could against the wall to try to find that cure. This happened to be one of the most active molecules that they studied. The other thing that was very interesting about it was, as they were throwing things against the wall looking for the cure in cancer, there's a little keyhole in that wall that's called the blood-brain barrier. Most things don't get through it. Your body's designed to keep things away from your central nervous system for good reasons. This was one of the few molecules that was studied that actually crosses the blood-brain barrier. So a fair bit of that work at the National Cancer Institute was focused on CNS, or central nervous system tumors, including glioblastoma. So there was evidence of activity against our target indication in the published literature that we can leverage. The business model is simply to take that, modernize it, focus it where it makes sense, and run it as quickly as possible. We understand the safety profile, we understand the mechanism, and interestingly, because this product is approved in China, somebody has already invested in a commercial scale-up, which we can leverage as well. And I'll talk a little bit about that. And of course, we filed new patents around this. So again, the business model is take all that wealth of information that's available to us, wrap new intellectual property around it, modernize the study and understanding of the mechanism of action, and plug that in where the mechanism of action says we can solve an unmet medical need. And the first place that we've decided to focus is as I mentioned a few times, glioblastoma. As many of you may know, GBM is a fairly large market, although it is an orphan cancer. About 15,000 cases a year in the United States diagnosed every year. Unfortunately, with all the advances that have been made in cancer in the last 20, 30 years, the outcomes with glioblastoma haven't changed very much. Without treatment, the median survival is less than 6 months, and even with today's standard of care, is less than a year and a half. This is a cancer that affects people in the prime of their life, in terms of people between age 45 and 60. So even though it's only 15,000 patients a year, it's nearly impossible to be in a room with more than 30 or 40 people where someone doesn't come up to us and say, somebody I know is either suffering from this or has died from it. In fact, somebody this morning already said that they knew 2 people. So that sort of validates that, unfortunately. Most of the patients fail the current therapies very rapidly, and there's nothing for them. And we believe that VAL-083 provides a potential answer there. So this is some of the original data that excited us in glioblastoma. What we're doing here is comparing VAL-083 to the current lead therapy in the market, temozolomide, which is marketed by Merck. Temozolomide, or Temodar, does about $1 billion a year in sales in glioblastoma worldwide. The challenge is it only works in about 40% of the patients. We now understand why. But comparing this current drug to the old data from VAL-083, what you see here on the bottom here is that we are comparing a published phase 2 clinical trial, a randomized clinical trial with VAL-083, to a phase 3 study with temozolomide, which was the same design, radiation versus radiation plus the drug, and look at the benefit of adding the drug to the therapy. And what you'll see here is that with temozolomide, the benefit was 2.5 months versus radiation alone. That is what drove this therapy into the frontline therapy for glioblastoma and $1 billion in sales. If you look at the published data from VAL-083, it was almost 8.5 months benefit versus radiation alone, which on the surface is, wow, that would knock your socks off. However, you have to sort of take all this with a little bit of a grain of salt in terms of time. The VAL-083 study was in 1979. The temozolomide study was in 2005. So the quality of the surgical intervention and the radiation delivery would have evolved in that period. And so the question is VAL-083 still statistically significant if you shift the curve over and delivered radiation surgery in 2005? And the answer to that is yes. The other thing that's important to look at is the overall survival of these patients. And if you buy the concept that the radiation and surgery improved between 1979 and 2005, which you should, the overall survival being comparable between VAL-083 and temozolomide in the same study design becomes very profound. So there's a lot of mental gymnastics that you can do with this data and the other publications that are in the literature. But to us, we were asking the question, does there appear to be activity with VAL083 in our target indication of glioblastoma? And the answer is clearly yes. And in fact, the overall survival and the outcomes are comparable to any other chemotherapy that has been approved in glioblastoma. So we said, okay, well, this is very interesting. We've got a drug that seems to work as well as what's out there. But so what? The so what is the mechanism of action is different. Temozolomide and the other agents that have been approved in glioblastoma, the other chemotherapies, all work the same way in terms of where they bind on DNA. There's some nuances to their mechanism, but they all attack the O6 position on guanine. What we knew was that VAL083 attacks the N7 position on guanine. It hits DNA at a different point. We found that to be very interesting, and it became much more interesting a couple of years ago when the mechanism of resistance to temozolomide and the other alkylating agents that work against that point on DNA became well understood. There's a repair enzyme that's called MGMT. It stands for O6-methylguanine methyltransferase. Its sole job is to fix damage at the O6 position on guanine. We all have it. It's supposed to be there. It's meant to repair DNA damage. Unfortunately, if you're trying to use an agent that causes damage at DNA to kill cancer, and you have a lot of that enzyme, which most of us do, those agents don't work well. And so we said, well, we're working at N7. We should be able to demonstrate that our agent is active independent of MGMT repair. And that's what this data is here. You're looking at a patient that does not express high levels of MGMT, and you'll see the blue line on the top here is temozolomide. As you go across the x-axis, you're putting more drug on the tumor, and you see the blue line dips down. That's called a dose response curve. The more drug you put on the tumor, the more the cancer cells are dying. The pink line is VAL083. So you see it drops down a little bit more quickly, which means that our drug in that patient, in that cell line, is more potent. That's very interesting, but not really the point. The bottom panel here represents most of the patients. 60 to 75% of those who are diagnosed have a high expression of that repair enzyme MGMT. You look at the blue line as you go across and put more drug on the tumor, it never dips down. It doesn't do anything because that repair enzyme is preventing the cytotoxic activity of tempo. The pink line looks exactly the same. We're independent of that repair mechanism, which does two things for us. Number one, it justifies going after our initial target market, which are patients who have failed other therapies, the refractory disease market, which gives us a very streamlined pathway into a registration trial and commercialization. The other thing that it does is it gives us a stepping stone to move this drug into the frontline therapy, which is the billion-dollar opportunity. The measurement of MGMT, that companion diagnostic that everybody talks about when they're trying to identify patients to target in a clinical trial and in the market, is already in place. The measurement of MGMT is a CLIA-approved diagnostic that can be used routinely in the pathology lab and is currently used as a prognostic to guide for indicating outcomes and basically sitting down with the family and saying, you know, your loved one has glioblastoma, the outcome and the likelihood is poor or really poor. But it's in place, and we can use that as a companion diagnostic as we move toward the frontline therapy in the future. The other thing that was interesting to us is we know what the NCI did. We know their dosing regimen. And we said, well, this drug is a little more friendly than other alkylating agents, a little more friendly than in terms of its toxicity profile than what the NCI assumed. The dose-limiting toxicity with all of these agents, all of the alkylating agents, whether they work at O6 or N7, is something called myelosuppression. It pushes down your white blood cell count. So you can dose the patient for a little while, and you have to wait a few weeks for the white blood cell count to recover, and then you dose again. What we knew was that the assumed waiting period, what's called the nadir and recovery, was shorter than what the NCI had assumed. So we decided we could probably dose more frequently than what the NCI did, hit the tumor more often. The other thing that we noted was that the only grade 3 and 4 toxicity that has been reported with this drug in the literature or in the overseas commercial experience is that myelosuppression. There's no cumulative organ damage, for example. So just because we can manage myelosuppression more readily and better than you could 20 years ago, we should also be able to push the dose higher. So we should be able to hit the tumor harder, more often. deliver more drug to the tumor more often, and hopefully have even a better outcome than what was very promising already in the hands of the NCI. And that's what this slide here demonstrates. Is the red here is the NCI's regimen. They would dose for 5 days and basically wait a month. They had a 33-day regimen. What we're doing is we're dosing 3 days out of every 21. Our target dose pushes the dose much higher than the NCI as well. So hitting the tumor harder more often will take advantage of both the higher Cmax and a higher area of the curve, a higher exposure as we deliver this drug against the tumor. So based on that strategy, we initiated a phase 1-2 dose escalation trial. We are treating patients that have not only failed Temodar, but have also failed Avastin. Avastin was approved in 2009 for the treatment of refractory glioblastoma on an 85-patient open-label study with less than a 20% response rate and a duration of response of less than 4 months. So the bar has been unfortunately set very low. This is a very serious situation, and Avastin was not shown to impact the survival of those patients at all. So as we go forward, we'll be looking at a relatively small open-label registration trial. We have currently 30 clinical sites, UCSF in California, as well as Sarah Cannon and the affiliate Sarah Cannon site in Tennessee and in Florida. We've been presenting data along the way at major cancer meetings, and we'll continue to do that, and I'll show you a little bit of that data in a second. So the idea is we'll test our new regimen at a lower dose and make sure it's safe and move up as we go forward. And eventually, and actually very shortly, we'll be above the dosing level that the NCI was using, and I'll talk a little bit about that in a second. Now, the FDA had asked us to start at a relatively low dose. We did do that, and we've been successively moving up the ladder in terms of the overall dose and exposure. We have recently completed the 20 milligram dose, and what I'd like to point out is the cumulative dose in a comparative 33-day cycle. The National Cancer Institute's dose in the 33-day cycle is 125 milligrams per meter squared. The dose we have just completed is 120. We will be announcing shortly after a mandatory safety waiting period that we are moving to the 30 milligram dose, which means we'll be delivering 180 milligrams per meter squared in that same window. And our target dose delivers 240 milligrams per meter squared, comparable to the NCI's window where they were delivering 125. So again, hit the tumor harder more often. The NCI was seeing response rates on the order of 40 to 50% in various publications against glioblastoma. Our goal is to show that this drug works the same way it did 20 years ago in our target population of refractory glioblastoma and move it toward commercialization as quickly as possible. And remember, the Avastin approval was based on a 19.4% response rate. Our results to date are that we're obviously seeing no dose-limiting toxicity so far, so safe and well tolerated. A portion of patients, even at the lower doses, are seeing a response in terms of radiographic imaging and the tumors are shrinking. So one of the things that's important to keep in mind here is that the R-squared, the link between progression with these patients and death is 0.98. So if you can halt the tumor's growth, or better yet, shrink it, the patients tend to survive. And that's what we're seeing in our study, even at these lower doses. So we're excited about what we're seeing so far and looking forward to moving into higher doses. The pharmacokinetics, the PK with this is exactly what you would expect if you looked at the PK from the literature and made a model of the exposure that we would expect to see in plasma. We're seeing exactly the same thing in our study. It's like bang on where you would expect it to be. The drug readily crosses the blood-brain barrier and has a long half-life once it gets there. And it's been shown to preferentially sequester tumor tissue versus normal brain matter. So everything that you would want to see in a small molecule therapy against glioblastoma is the profile of this compound. As I said, we're seeing some evidence of responses in the early doses, which is exciting to us, and we expect that to improve as we get to higher doses. We're not only seeing radiographic response, but clinical signs as well. When the tumor shrinks, less pressure on the surrounding tissue, people's speech is able to improve, so because their use of their arm comes back. It's a very profound and durable clinical signs in addition to the radiographic response that we've observed today. Now, the FDA had asked us to take what we would consider baby steps in the dose escalation, and the reason for that was was very logical. The dose-limiting toxicity with our drug is expected to be related to white blood cell suppression, including thrombocytopenia or platelet suppression. We're following Avastin, which has a well-established bleeding risk. So even though we're washing out Avastin out of the system before we start, it was recognized that there were some phenotypic changes to the tumor and the body following Avastin therapy. And nobody really knew whether or not coming in with a drug that may suppress platelets would cause problems. So we started at a relatively low dose. We didn't see any evidence of these types of problems. And we're also seeing some evidence of activity. So last summer we went back to the FDA and said, let us go faster. Let's skip some of these baby step interim doses. Let's go back to a more traditional dose escalation and get this drug to the point where it should be moving into a registration trial as quickly as possible, because this is a huge unmet medical need and we think that we can benefit these patients. And the agency, after a pretty thorough safety review, agreed with us. So we've been able to accelerate our dose escalation and take many patients and dose steps out of our clinical trial, which will save us about 40% of the time and money to get from where we were into the registration trial. So our goal this year is to complete the dose escalation study and move into that registration trial in the second half of the year. Where we are with that process, as I said, we are finishing up a safety waiting period on the 20 milligram dose. We anticipate announcing moving to the 30 milligram dose or the next dose relatively shortly. We anticipate presenting data at AACR, the American Association of Cancer Research, in April in San Diego. Also anticipate presenting data at ASCO in Chicago in June, and hopefully be in a position to have interim data from that registration trial at the Society for Neuro-Oncology meeting in November in Miami. So again, communicating with the market as we go forward and the medical community as well. Now, as I mentioned in the preamble, this drug is actually approved in China. We didn't start out looking for a Chinese drug. We were focused on the data from the National Cancer Institutes. What we needed to do was find somebody to make the drug product for us when we decided to start our own clinical trial. And this is a cytotoxic, so at an early stage, you typically would go to Eastern Europe or China or India to make the white powder that you would eventually create your drug product with. And what we learned as we were going through that process was there was somebody in China who not only was making the API, that white powder, but they were actually making drug product because the product is approved in China for CML and for lung cancer. So we said, okay, that's interesting, but we're focused on glioblastoma, so let's put that on the side for a second and ask the question, is this drug product and is this manufacturer of the quality that we could use in our clinical trials, and could they potentially be a commercial supplier for us down the line? The answer to that was yes. So we established an exclusive relationship with that supplier. We've also now looked at how this drug is positioned in the marketplace and believe that this could be positioned in a better way and the sales could be grown, and we would look at potentially taking a royalty on improving those sales. CML is a relatively small indication in China, but lung cancer is a very large indication in China. It's expected to be 1 million cases a year by 2025. 90% of those will be non-small cell lung cancer. All of them get the platinum doublet therapy. So our question is, how does this drug stack up vis-a-vis platinum therapy, and is there a place for it in the marketplace that it could be better positioned than it is today? And the answer, we believe, is yes. This is some data that we presented at a meeting in Boston in the fall. And what we're doing here, it's a very simple animal model where you're taking a mouse, establishing a tumor in the mouse, treating with a platinum agent, treating with OA3, and treating with the combination and seeing what happens. Everybody knew that platinum worked relatively well in this model, so we were pleasantly surprised to see that VL-083 actually works better in this particular model. And the combination of the two was better than additive. So that was exciting to us. It gives us the opportunity to move into more aggressive lung cancer models, and we anticipate presenting that new data at scientific meetings as we go forward in the spring. But it also establishes what we believe is a strong position for the drug vis-a-vis platinum therapy in refractory as well as newly diagnosed non-small cell lung cancer. So we've established a network of clinicians, not only in lung cancer, but also in CML and in glioblastoma in China, who can help us to leverage the opportunities there. And we anticipate conducting some initial clinical studies, validating clinical studies, particularly in non-small cell lung cancer, with those key opinion leaders. The idea is a phase 4 program that is market-driven, publication-driven to reposition and grow sales in China, also looks very much like a phase 2A type of program for the rest of the world. DelMar will be responsible for conducting those studies, and our manufacturing partner will pay for them, which means that the data that comes out of that can be created on somebody else's nickel and usable by us to establish a new indication as we go forward around the world. Our goal this year is to initiate those clinical studies. We have established relationships with the KOLs. We are developing protocols there, and we anticipate doing that in the relatively near future. And to establish a partnership with someone who has an oncology sales force on the ground already in China, because we're not going to have our own sales force. Our manufacturing partner does not have an oncology sales force, and our goal is to simply provide data to reposition the product so somebody can sell it effectively and take a royalty on sales. One of the other questions that everybody asks is, hey, this is an older drug. It was studied at the NCI in the 1970s and early 80s. How do you protect the intellectual property? Well, first of all, we have orphan drug protection in glioma in the United States and Europe. So even if we had no new patents, that would give us market exclusivity for 7 years here and 10 years in Europe from the point of approval. We have also filed 7 new patent families, and the first one of those patents is issued in the United States last summer, which gives us patent protection here in the US through 2030. And we're, of course, prosecuting all of those around the world under PCT. This patent strategy has been well validated. It's essentially the same way that Solmetics protected a drug called bendamustine. Bendamustine was around in Eastern Europe for 35 years before Solmetics picked it up and developed it. It now does about $750, $800 million in sales in refractory non-Hodgkin's lymphoma primarily. Very similar business model as well. Older asset, look at the mechanism, plug it in where it makes sense, and we believe that that strategy saves a lot of time and money and has been shown to be effective many times over. My background is biophysics. I spent some time in the benchtop. I was at KPMG in the Health Ventures Group before being recruited by some investors that we had known through clients to help turn around a company a few years back. I've also started a venture-backed company with 2 professors from the University of British Columbia, raised about $40 million through Series A and grant funding, including from the Gates Foundation. Built a team, took something that was from an interesting biological observation into the clinic. And I was introduced to Dennis Brown in 2009 as he was exiting a company called ChemGenics. Now, ChemGenics, again, is the same business model. Taking an older asset, in that case, a drug called Cinrybo, which is now marketed by Teva, was shown to be active in patients with the T315I mutation, which failed Gleevec. This was a huge unmet need, and Cinrybo solved that problem. That company was acquired in 2011. As that happened, the key members of the team spun out, and we formed DelMar around the next asset that Dennis had identified. When that team's all around the table, we have about 20 oncology approvals under the belts of the various people around the table. So this is a team that's been successful in drug development, successful together in building a company, and successful in exits under this business development and commercialization strategy. Around us, we have a great group of advisors. Bill Garner is our co-founder as well. He's actually been responsible for introducing Dennis and myself a few years back. John Bell is runs a fund that invested early with us. Rob Toth, some of you may know. He was the analyst at Prudential for many years before retiring, and he's been a friend for a long time. Victor Levin is the individual who actually sourced this drug originally from the NCI, when he was head of neuro-oncology at MD Anderson. He still treats patients in Redwood City, California, which happens to be next door to Menlo Park, which is where our clinical team is located, so he's been actively involved in the project. Susan Chang is head of neuro-oncology at UCSF, one of our clinical sites, and probably one of the most well-known and most important clinical researchers on the West Coast. Jim Perry's the head of the Canadian Brain Tumor Consortium. That work that elucidated the importance of the MGMT repair mechanism, Jim Perry was the lead author on that work, and he's been involved with us from the beginning. Skip Burris was our first clinical investigator. The team's had a long history of working closely with Skip. Bill Bedell works with us on the mechanism of DNA damage and repair. Dan Zhang, some of you may have met. He's a member of the Chinese FDA Oncology Advisory Panel. So as we go forward and do clinical studies, one of the things in China, one of the things that is important to us is to operate in the context of the current label, so we're not going through and applying for permissions from the agency, so Dan gives us some good advice there. Christine's an analyst who retired a number of years ago from up in Toronto, and she's probably one of the toughest people on the street. I've known her for a long time. We've become friends. I know she's made lots of CEOs cry over the years. One of the things that we did is we had our business strategy and thought that all our ducks in a row, and of course we were drinking our own Kool-Aid, so we sent everything to Christine before we started the company and said, tell us what we're doing wrong, and she's great at doing that, and we continue to interact with her to sort of make sure that we don't lose sight of the realities. And of course, Solvair is pretty much the architect of taking an old drug that nobody wanted and building a company around it. Sol took a drug called thalidomide, which many of you know had been pulled from the market because of some horrible side effects. And when Celgene was in its early days, they were actually a client of ours at KPMG. And we said, well, this drug should be useful in a rare form of cancer. And everybody thought he was a little bit off his rocker, but what he did is he showed that was true based on the mechanism, brought it forward, got it approved, and built a company around it. Most of the companies with our business model are acquired before the drug is approved. Chemgenics, for example, for $235 million before the approval of Cinryvo. But Celgene's an example of commercializing this and building a company around it. And several tens of billions of dollars later, obviously Celgene's been a huge success, and if we could have a fraction of that, we'd be certainly very pleased. So our history and all the things that we've accomplished since we started, hopefully convince you that we're a company worth watching. We are on the OTC, and as a new public company, our visibility is not necessarily what we think it could be. We're working very hard on that. We priced our round in our trading compared to our peer range. We think that as we move toward more senior exchanges and the visibility improves and the ability for traders to actually work with our company improves, that we will hopefully trade up into the mid-range and beyond versus our peer companies. So thank you very much.